Good day, Homo sapiens. Welcome back to studying Prescott's Microbiology. And finally, today we are at Chapter 2. Chapter 2 is about the study of microbial structure, microscopy, and lenses. And before going to that, let me just first check if my live is working fine. I need to know that because it will be in vain. And it's my routine to do this every day, so you can just skip it. Yep, it's working fine. So let's leave it at that. Now, let's talk about this chapter. So, the study of microbial structure. This is not explained in this chapter like completely like the study of prokaryotic cells and eukaryotic cells, etc. But how do we study this microbial structure? This is explained in this chapter, and that is by microscope and everything, starting from the lenses. Look at this, lenses to the working of microscope is told to us. I think when we study microscope, we don't usually go in detail with about the lenses, so I think this is a good start. And to be honest, I suck at studying the lenses, right? Like, I don't know. I am like you in the field of microscope, so let's study together as I mentioned in the title of this video. So this is all that we will be covering in the chapter. I think I just slide it through so you can see what kind of stuff we are going to study. So, but today, by the way, brace, okay? I think this chapter is going to be a complex one, especially with me telling you guys, because I have I'm not that bad, but let's start. Let's see how much I know and how much this book knows. Let's see how much we're gonna learn. So, starting from the introduction is always good to set up the mood, right? And let me enlarge on this first, and let me check if everything is okay. Everything's okay. So we are starting with introduction. Do we study what we are going to study in this chapter? I usually don't like to read this. So light microscope, many types of light microscopes like bright field, dark field, phase contrast, fluorescence. Okay, there are multiple types of light microscopes. Then bright field microscopy, magnification of light microscope, electron microscope, new forms of microscopy. So we are going to study that, right? We don't have to go into the detail. By the way, guys, I didn't finish the first chapter, like the future of microbiology, and I just jumped into that because I was so interested in reading this. So we will go back to that future of microbiology in another live or in another video. So sorry guys for stalling you, but now we are starting it. Microbiology is usually is concerned with the organisms so small that they cannot be seen distinctly with unaided eye. Unaided eye means not your usual eye, the eye that is being helped with something else. Like if it is not being helped with something else, you cannot see it. Because of the nature of this discipline, the microscope is of crucial importance. Yeah, this is the nature of our discipline, that we cannot see the organisms, but we want to study it, but we are going to study it. So microscope comes, as a important aspect of this microbiology. Thus, it is important to understand how the microscope works and the way in which the specimens are prepared for examination. Like if we know how the microscopes work, that's how we are going to make our specimen perfect, right? You guys will be thinking like, how is it difficult to make these specimens, right? It's not that difficult, but it is, because usually people make bubbles in their slides, or you can say they make their specimens very thick or very thin, or they don't color it right. Like there is a lot of things in you preparing the specimen. So in this chapter we begin with the detailed treatment of the standard bright field microscope, and then describe other common types of microscope. They are talking about, we begin with, they are lying, they begin with the lenses. Okay, sorry, I didn't mean to say lie, by the way. So they are starting with the lenses, not directly the treatment of the standard bright microscope, then describe the other common types of light microscope. Next, we discuss the preparation and staining of specimen. Okay, they are talking in general. So first we study how the microscope works, then we study how the staining means coloring of the specimens, because microorganisms are usually almost invisible if you remove the fungi, right? Fungi are usually colorful, but no, wait, they look colorless under the microscope as well if we don't stain them, right? Although they are very much easier to study in comparison to bacteria. So this is followed by description of transmission and scanning electron microscope, both of which are used extensively in what in current microbiological research. Yeah, if we advance, there are even smaller microorganisms we have to see, or there are microorganisms in which there are even smaller organelles in them, or even smaller structure in their cells that we need to use my electron microscope to study them. So they are saying we use electron microscope. By the way, electron microscope is so expensive. I think it is usually 1 or 2 in a specific area, unless there is a big research centers or things like that. So we close the chapter with a brief introduction to the 2 newer forms of microscopy: confocal microscopy and scanning probe microscopy. Oh my god, probe. Okay, let's not talk about this, like they are going to explain it in the last. So looks like we are going to get a lot of things done in this chapter. I'm really excited about this chapter because this is so interesting. Lenses and the bending of light, right? What do we use lenses for? Yeah, to change the trajectory of the light, right? The concave and the convex lenses both change the light, not like they don't leave the light in a straight form. To understand how light microscope operates, one must know something about the way in which lenses bend and focus light to form images. They are saying if you want to know how the light microscope works, you have to know how the lenses work and how do they focus the light to form images. Yeah. So they are saying start with the lenses. When a ray of light passes from one medium to another, refraction occurs. We have studied this. And those who didn't, medium means, there are 3 types of medium in our world. That is air, water, and solid, right? They are saying if in any case the the light changes from one medium to another, like from air to water or water to solid or solid to air or things like that, the refraction occurs, means the light bend a little bit, differently, like the angle changes, but it still passes through it in the straightforward way. That is, the ray is bent at the interference, right? The light bends from which area? Interference means, like, this is a line and the light came from here, right? I can't make the arrow. Wait. I do have this kind of thing. Like this is one medium. Sorry for my crappy drawing. This is another one, right? So this is and there is the light coming from, right? Okay, why am I making the light so wrong? So this is the light that came, right? When it strikes another medium and enters into another medium, it slightly bends like this, or maybe like this. It depends on which medium it is changing into, right? If it is denser, it refracts more. If it is not denser or lighter, it refracts into, more like this angle, like the angle changes. So it can be this way, pencil bends in the water as well, right? So this is the angle. Like this was the original angle and this was the angle afterwards. And they are saying, okay, so this is the line and that's how light entered and this is how it bent. So the point where it bended, it is the interface, right? Yep. So, and what again? When the light passes from air into glass, solid, they mean. A medium with greater refractive index, it is slowed and bent towards the normal line perpendicular to the surface. So a medium with greater refractive index, like I said, it depends on the medium. It's because of their particles, right? Again, should we go? Let's make it a little bit smaller so it doesn't go up. Oh, what are you doing? Right. All right. By the way, why do I have to show? They are saying no. All right, so they are saying, like I said, it depends on the medium particles, right? Like if this is solid, that's how the particles usually is, right? That's how it is. Like they are so close or packed. I still left the spaces in them. They don't have spaces. But if you talk about the air, the particles are like this, right? So when the air that is going to this, they are saying that the refraction rate is greater in this. If you compare it with the liquid, right, their particles are less packed than the solid one. So they are saying the refractive index of the glass, or aka the solid, is higher than the air. So it is determined by the media, right? So they are saying that if it goes from lower, you can say lower refractive index medium to the higher refractive index medium, the light speed, right? The speed of light slows down. Okay? So the speed of light slows down. And what other thing are they saying? That is, the ray is bent into the interface, bent at the interface, the matter how greatly a substance. What am I saying? The refractive index is a mere, guys, wait, I need to change it. I can't read it. I know my speed is slow, but guys, we are studying this. If you are here to think that you are going to take a quick explanation, you are in the wrong place. Kindly go and watch some quick videos. We are here as an activity, right? We are studying as a fun, and we are gaining knowledge in that. So the direction and the magnitude of the bending is determined. Oh, no, I think we left something. Is a measure of how greatly a substance slows the velocity of a light. Okay, so they are saying the refractive index is a measure of what? How the velocity of the light slows down, and how its direction changes, and how is the magnitude of bending is done. So is a measure of the substance, how the substance slows the velocity of light. So how is the velocity of light slows, right? It is determined by these, the direction and the magnitude of the bending. And it is in turn determined by the two medias forming the interfaces, right? The bending of the light, the angle, the direction, like it can bend this way, it can bend this way, and it can bend that way, right? Three kind of ways. It is determined by what is the medium. If the medium that is it is being entered from has lower refractive index than the one that is being entered, then it will be going to go slow, right? It isn't going to go faster because the particles compact and the light haven't have a way to move freely in that. So how did they show this? When a light, okay, I left that. No wonder I thought, where was I going with that? When light passes from air into glass, a medium with greater refractive index, it is slowed and bent towards the normal. Guys, this is what is this? So this is how the normal should go, right? It is going to go towards the straight line. All right. Here, like if the light would have been in the same medium, it would have moved this way, right? It would have moved this way. But if you look at the angle that it made when it was bending, like this and this, like we may have bent angle from this side. So the way it is bent is towards the one that is, the dots, where the dots are, and this is known as the normal line, right? This is normal line. And if you look at it this way, again, when it changes from the same medium into the same medium where it came from, again it changes away from the normal line. Look, isn't it changing away from the normal line? Like, it is the opposite scenario, like, right? If this medium is denser, this is lighter, right? If it goes from the lower refractive index medium into the higher one, let's make notes. Lower into the higher, right, refractive index, bend towards normal, right? And if it is going from higher into lower refractive index medium, then it is going to bend. How do we draw away from normal? Away and towards, right? I hope we get this point. Till this point we get it, but I think we still don't get it, right? How this normal line is formed actually. And the angles, I can get it. That theta, like it I think it is the angle, this angle. In this case it is this angle, and in this case it is this angle. They are making an angle which is, in relation to this line, right? And in this case the theta is this one, and in this case the theta is this one, right? So we get this point that when it enters from lower refractive index medium into the higher refractive index medium, it changes towards the normal, right? The light bends towards the normal, and when it leaves the enters into a lower refractive index medium, then it bends away from the normal line. So here it is, guys. Look, let's read this. It is slowed and bent towards the normal line perpendicular to the surface. All right, that's what I was asking. Like, how did they draw this normal line? And they are saying that they made it. Wow, I can't do anything without this pencil now. Right, they are saying that this is the medium, and they made the normal line away from it, right? Like perpendicular. If you look at this is the 90 degrees, right? If you look at this one, right, this is 90 degrees as well. If you look at this one, this is 90 degrees as well. And if you look at this is 90 degrees as well. If you look at from this side or that side, whatever side, it will make 90 degrees. So they are saying that so they change it, right? And bend towards the normal line perpendicular. As the air leaves the glass, it returns to the air a medium with a lower refractive index. Where is the normal one written? Where is it? It accelerates and it bends away from the normal. All right. See, the opposite scenario. If it is entering a higher refractive index medium, it slows down. And if it is doing the opposite thing, it's going to accelerate, right? We get it now. And thus a prism bends light because the glass has different refractive index from the air, and the light strikes its surface at an angle. Right, they are saying why when we look, like when we pass the light from the prism, guys, you know what prism is? This is prism, right? No, by the way, it can be from any shape. Prism is the material that is a glass material that changes the light from one wavelength into 7 wavelengths, right? Red, blue, yellow, vibrio, right? So no, not vibrio, what was that? Webgoer or something, like indigo, blue, violet, red, orange, yellow. So those kind of wavelength it changes into. But if you look at this, when it enters into that prism, it never ever come out like this. It doesn't go this way. It bends here, and then it bends a little bit and go this way. If you have ever experimented with the prism, you would know that that the prism refracts the light in a different in a different angle than how it entered into the refractive in the prism. So now what happens? Lenses act like a collection of prisms operating as a unit. Yeah, so they are saying that lenses act as the prism, but lenses, is a collection. There isn't a one microscope in one lens in the microscope. There are multiple, and they all act together to work as a unit. When the light source is distant so that parallel rays of light strike the lens, a convex lens will focus these rays at a specific point, the focal point. They are saying when the light source is distant mean they are, there is a quite gap in the lens and the light, so that the parallel rays of the light strikes the lens. Here I think we don't need to draw it. Here it is. These are the parallel rays, right? Parallel rays. So when it strikes the lens, I think they use the convex lens in the microscope, right? They are saying they bend these light all into one point, and that is your focal point. This point is your focal point. Do I have to go through this every time? Do I have to erase this every time? So a convex lens will focus these rays at a specific point, the focal point. The distance between the center of the lens and the focal point is called as focal length. Guys, in microscope, look at this. How you might have a question in mind, like how do you know if this is the exact alright, my OBS was disconnected. So we need to make the lens work this way, that it should bend the light and make the focal point on this area. No other area than this should be the focal point, right? If the focal point was in the center, like this area, right, this area, and not this area, so where is our specimen going to be magnified? It can't be magnified if we don't put the light into a focal point. So the distance between the center of the lens and the focal point is called as focal length. They are saying this area, this as is shown, that this is the focal length. Alright. Now, our eyes cannot focus on object nearer than about 25 centimeter or 10 inches. Couldn't they tell us into the millimeter language? How much is 25 centimeter? Our eyes cannot focus on an object nearer than 25 centimeter. Guys, I have to search this. Definitely have to search this. 25 centimeter in millimeter, right? 250 millimeter. How much is 250 millimeter? And I need to visualize it, right? Where is the scale? Alright, guys, bring your eyes closer to this distance. And yeah, it's true, I brought my eyes closer. It doesn't work, it doesn't focus, it makes a strain on your eyes. 10 inches. No, wait, 10 inches? 10 inches way too far. I can focus. Guys, I can focus on 10 from far away of 10 inches. I haven't studied. Alright, on objects near. Objects, I think objects needs to be larger than 10 inches in that case. But wait, I can. How much is 10 inches? This much? Guys, I think I can focus further from 10 inches. Whatever, if they say it, say it. No. Why will I believe this? Till how much can our eyes focus? Look at this. They are telling me myopia. Let's see how much they. What the heck? I wasn't asking this. Typically, near point of your eye, typically around 25 centimeters. So they are saying the same thing. Why are they saying the same thing? And let me, by the way, let me bring this down, otherwise we cannot see. The limitation may be overcome by using convex lens as a simple magnifier or microscope, or holding it close to an object. They are saying, how can we overcome this limitation? How can we focus further than this point? It is by using a convex lens or a simple magnifier, right? And holding it close to an object. A magnifying glass provides a clear image at much closer range, and the object appears larger, and the lens strength is related to focal length. A lens with a short focal length will magnify an object more than a weaker lens having a large focal length. Alright, how does it magnify? They are saying if this focal length is short, that means this lens is weak. If this is longer, that means that this lens is stronger and it can magnify an object more. Why? Why is that? No, like why? Are they going to explain to us? I thought this was going to be difficult. It isn't that difficult. No wonder I took my time. Okay guys, what is this? Common units of measurements. We know the micro, nano, millimeter, centimeter. Guys, we know it, right? We don't have to explain it. And we know that this centi is 10 raised to the power minus 2, milli minus 3, minus 6, minus 9, and angstrom minus 10. Guys, we know this. I am pretty good in physics prefixes. These are prefixes, right? The centi, milli, and micro, nano, because I memorized it when I was in 10th. You guys might have done as well. Who knows? So not might have, you should have, because in the exams that comes. So this is the light microscope. Now guys, do we understand the previous concept? We just studied the refraction. We didn't have to do anything with the reflection or the diffraction, so we didn't study that. We just studied the refraction, and we just studied the simple things, and we just studied that like how the light slows down, how the light accelerates, how in which direction does it bend and in which direction does it not bend. We studied about the focal length, and we only talked about the convex lens and not even the concave lens, right? And they are saying the larger the focal length is, the better it is. The smaller the focal length is, the weaker the lens is. Guys, but I do have question. And we are going to ask that question. And that is, why is lower focal length convex lens weaker than so let's see, is considered stronger than the larger focal. What the heck? Oh, sorry, I mixed it. So the lower it is, the better it is. Then I get it. Right. Because shorter focal length means the lens bends light more sharply, causing greater convergence and therefore a stronger. Guys, sorry for my, for my weak brain. I completely forgot they were talking about this small. For smaller I get it, like for longer I didn't get it. Like, if it is near, then it will bend more sharply, like, this is bent like this way. If this focal length was here, this focal point was here somewhere, like, So of course the light wasn't going to go bent that much. If you bring the same focal point here, it is bent more. Like look at this angle, it's quite a lot of a difference. So I get it. Shorter means stronger, longer means weaker. If I was saying it the wrong the whole time, forget it. Finally, we are going towards the light microscope. Microbiologists currently employ a variety of light microscopes in their work. Bright field, dark field, phase contrast, and fluorescence microscopes are most commonly used. Modern microscopes are all compound microscopes, that is, the magnified image formed by the objective lens is further enlarged by one or more additional lenses. They are saying that the common current microscopes that we use are the compound microscope. That is, that the magnified image formed by an object is further enlarged by one or more additional lens. Yeah, like we can't do anything with the one lens, so if we can do things with one lens, we have to use the multiple lenses. The bright field microscope. Let me guess before going into that. I think that by bright field means, the slide we use, it has a background, right, when we put the specimen on it. So I think if it is the transparent one, it is the bright field microscope, and if it is the darker one, it is the dark field microscope. This is how much I am getting, right? Let's see if I'm wrong. The ordinary microscope is called the bright field microscope because it forms a dark image against the brighter background. Let me close this. Some people might not like the darker, The ordinary microscope is called the bright field microscope because it forms a dark image against a brighter background. Yeah, the background is brighter. The microscope consists of a sturdy metal body or stand composed of a base and an arm to which the remaining parts are attached. We already know it is a metal body and it is a stand composed of a base, right? Like I don't have to draw it. And an arm to which the remaining parts are attached. Guys, what is arm? Do you know? What is the arm? Let's go down. So this is arm. So they were talking about this is the sturdy metal body, and this is the base. This is the arm, which has its head, and it has the remaining parts attached to it. A light source, either a mirror or an electric illuminator, is located at the base. So guys, we usually think that the light will go from, the upper area and directly towards the lens. But in microscope, it is different. Don't forget about the microscopes that you used in your 9th class, 10th class, or 8th class in your lower classes, which use the light which use the light from the environment, right? The natural light, and of course it came from the upper or from the sides. But the advanced ones, the light microscope, usually use the light from the bottom, and it then goes projected towards the slides. You can say the lenses. And the lenses, when we put our eyes towards the lenses, we can see the changed light. So a light source, either a mirror or an electric illuminator, is located at the base. Two focusing knobs, the fine and the coarse adjustment knobs, are located on the arm and can move either the stage or the nose piece to focus the image. Fine and coarse adjustment knobs. Coarse adjustment knobs, I think it is coarse, right? It is for the light, for our eyes, and the fine one, I think it is for stage. We have to look at this. This is the fine. Oh my god, I used the microscope, but why can't I remember which one was which? What is coarse adjustment in microscope. What? Oh my god, sorry, I was wrong. I'm always wrong. Coarse is for adjusting the range and fine is for the or eye adjustment, right? Like how it blurs and how it is, sharp. And they are both located on the arm and can move either the stage or the nose piece to focus the image. The stage is positioned about the halfway up the arm and holds the microscope slides by either the simple slide clips or the mechanical stage slips. Right. So they are saying the stage is positioned about halfway up the arm. The arm started from the base up till that, but the stage is in the middle of that, and they're saying that there are microscopic slides. It doesn't mean that the slide size is microscopic. It means that the slides that are going to be studied under the microscope. So that's why it's microscope slides. They are saying that can be either simple slide slips, the one that is sold in the boxes, or you can say mechanical stage clip. Like if there is the clip that comes with the microscope that you are using, right? You have to clean it, and here a mechanical stage. Okay, so this is clip, right? Clip. But this one is just the stage. A mechanical stage allows the operator to move a slide around smoothly during viewing by use of stage control knobs. Alright, so one is fixed and one is not fixed, right? They are saying, I think the one we used was the mechanical one, because we could move the slide, right? Like the one that is in our lab right now, I think it is the movable one, while the one that we use in our childhood, I think that were one of the fixed ones. So a mechanical stage, because we couldn't change it, right? Wait, let me, I needed to cross out that add is everywhere. Yeah, so it's because we can adjust it freely. So the mechanical stage is more proficient, you can say it is, but there are some specimens that you don't need it, that there is a need to not move it, like that one is better for the other one. The substage condenser is mounted within or beneath the stage and focuses a cone of light on the slide. A substage condenser is mounted within or beneath the stage and focuses a cone of light on the slide. Right, like we don't want, I think this condenser, this is E1 in electron microscope and this is E1 in the light microscope, both for the reason so that we don't want, unwanted magnification of other objects. We don't want our lights to go all the way around, and so our lens can't even know what light. It's not like it's a thinking object, but it doesn't know like what light should he focus, or like there is kind of error in that. It is not clear, so we have to condense that so it is focused on one point, and it makes it even easier for the lenses to change that, right, and make even a clear, sharper point and magnification. Its position often is fixed in a simpler microscopes, but can be adjusted vertically in more advanced models. Right, they are saying if simple microscopy, it is fixed, like we cannot change how much it can narrow down or how much above, or within or beneath, right? Like how we can change the location, we can change how much it condenses light, but they are saying in advanced model we can adjust it, right? Vertically means you can move it up and down. The curve, and you will know that if you move it up or down, the light is going to change. Let me show. Let me use my beautiful drawing skills. Like this is the light, right? This is the light that is coming from the bottom, you can say bottom of the microscope, and this is the condenser, right? Like we need this condenser, and this is the slide. They are saying that it can be fixed in the stage or it can be beneath the stage. You have already seen the stage, but let's forget about the stage. Stage means the slide, like the specimen we are going to study. So this light, if that went directly here, don't you think, like, I am very rough at drawing it, but it's not that bad actually, but you wouldn't, you see, like, it isn't focused and the lens doesn't know how to work, like if there is a lens in between there, right? So there comes a condenser, and this is a condenser. Like, it changes all of this light and it only goes here, right? Like this much. This is how it works, and the lenses just focus it more, you know, like focus on the object. Why am I drawing it so roughly? It's more like I'm making you forget what you studied. So this is how the condenser works. And if say there is a lens, by the way, lenses are on the upper edge. What am I doing? This is the, and you know this, there is a lens, right? Wait, let's forget it. There is a lens, and this is the light that went through it. So it's going to focus it more, like here. Wait, why am I drawing here, 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 here? Like, so they are going to focus it more, and we know that the smaller the focal length, the better it is, the better the magnification is. So that's how, oh, I wanted to show you how the narrowing work, the condenser works. So this is the condenser farther. They are saying that it can be moved vertically, right? Like up and down. You can move this condenser up to this area, and you can move this condenser up to this area, while the light source is here. Still the light source is here, right? Like, normally the light is going this way. Think about this. Think about this. Which condenser will have more light? Of course this condenser, because there is a large amount of light that is here, right? So the light is this way, right? While in this one the light is more narrower, and this one even more narrower. You know what I'm saying? Like, this is more condenser light, and these are, you know, like splattered across all the areas. So the curved upper part of the arm holds the body assembly to which a nose piece and one or more eyepieces or ocular lenses are attached. The arm that we have seen, right? This was the arm. There was the stage. So they are saying that the upper curvature of the arm, this is what they are talking about, holds the body assembly to which a nose piece means like where we put our face, our nose and our eyes align to see into the lenses. Into what? Into which we can look at one or more eyepieces or ocular lenses. You can call them eyepieces or you can call them ocular lenses. It's the same thing. It's the lenses. Ocular means from the eye, right? More advanced microscopes have eyepieces for both the eyes and are called as binocular microscope. Okay guys, let's see if this diagram, yeah, this is binocular microscope because it has two lenses for both of our eyes, while there are microscopes that has one of this, right? We can see with only one eye. Right. The body assembly itself contains a series of mirror prisms so that the barrel holding the eyepiece may be lifted for ease in viewing. Now they are saying multiple mirrors. Mirror is just for the adjustment of the light, right? Like, if you don't have mirrors, how are you going to see, further? Mirrors just reflect the image as it is. It doesn't change anything. So the image that you have gained from the lower lenses, it doesn't change. It comes the same way, but you can, make the lenses near to your eyes, a little bit further to your eyes, so it doesn't change. So the barrel holding the eyepiece may be lifted for ease in the viewing. Yeah, it can be. Like, it can be taken out, right? The nose piece holds 3 to 5 objective lenses of differing magnifying power and can be rotated to position any objective beneath the body assembly. Guys, this is what they are talking about. Here, this. Look, this is nose piece. What? So why they call it nose piece? Like this is our eyes, and there our nose is supposed to be here. Is it the nose of this arm? I don't know why they call this nose piece, but whatever. These lenses that are actually the heroes of these microscopes, their working matters the most. So they have different levels of magnification, and by magnification they means that these convex lenses have different focal lengths, right? So the one that would have higher focal length, like larger focal length, would be the weakest, means the magnification of that would be the smallest. Well, the one that had the lower focal length, that would be the magnification would be higher. So they are saying that the nose piece has multiple magnification lenses, and you can choose whatever you want. You can choose whatever you need according to your sample, like how much magnification do you need. Now you will be thinking like, isn't it better if we use the higher magnification from the start? So the answer is no. Sometimes you don't want to dive that much deeper into the specimen. You just want to look at the outer appearance of the specimen, so you don't need that much focal length. It will actually distract you from looking at the original structure of the specimen if you go with a higher magnification. But sometimes you do want to look into detail of that specimen or things, so you do need higher objective magnification. So and they are saying that they can be rotated. Yeah, you can rotate and bring it the one that you want to see in above the sample. Ideally, a microscope should be parfocal. That is, image should remain in focus when objectives are changed. Yeah, they are saying how you should change these. They are saying when you are rotating, you should be looking into these lenses, and you should be changing this while your eyes are looking clear into this specimen, right? Like you should be continuously looking and then changing this, because you don't want and it will be difficult for you to bring out that clarity into your specimen if you don't correct it from the start. Parfocal. Guys, I don't know what the parfocal word means. I get the point, but parfocal means, I don't want definition. Please don't give me definition. Having corresponding focal points in the same plane. It can also refer to all right. Parfocal means that all of the lenses that you are using, usually there are only two lenses, right? That's work, like your eyepiece lens and your objective lens, right? They should be parfocal, means their focal points should be the same, right? Like if that changes, you can never see the specimen clearly, whatever you do. So you have to align their focal points. I get it now. The image one sees when viewing a specimen with a compound microscope is created by the objective and ocular lenses working together. We know light from illuminated specimen is focused by the objective lens, creating an enlarged image within the microscope. Right, the lens is above the specimen, the light is below the specimen, and the condenser is below the specimen as well, or you can say within the stage below the specimen. And the light from the base goes upwards, and it goes through the specimen, and it changes its direction, because the specimen has different kinds of density in different areas. Like even if you take bacteria, there will be some areas which is liquid, some areas which will be solid, like some will be more solid than the other, like organelles you can say. So the light changes its shape, and when that reaches to the lenses, of course it's going to capture it that way, and the image that it will produce will make us will clearly shows how this specimen is, but it will be enlarged image because we cannot see it without the aid of this lens. So the lens is above, and after that I think they fix some mirrors. Even if there is no mirror, that goes directly to the ocular lenses, and it further magnifies it because it doubles it, and it even makes it sharper. So the ocular lenses further magnifies this primary image. The total magnification is calculated by multiplying the objective and the eyepiece magnifications together. For example, if a 45x objective lens is used with a 10x eyepiece, the overall magnification of the specimen will be 450. I think this part is simpler because we know this is the objective lens, this was the eye lens. Here is gone, and that. If there is 45x and the ocular is 10x, like we have to combine them, and it is not adding, it is multiplication because it is doubling it. Multiply it and it becomes 450x. Finish. Great, guys, we are done for today. Should we read more or should we not? Microscope resolution. Should we study the resolution, or wait? Should we study the diagram first? Resolution is pretty much higher, and I think we shouldn't read it today. Yeah, I don't know what they put into the resolution kind of thing. Yeah, guys, we shouldn't read the microscopic resolution. We are going in for a big. Alright, but we should look at this diagram. I think we already know the microscope general structure. Like, these are ocular lenses, you can call them eyepiece. This is the body. Yeah, body. The whole body is body, and if you make them in parts, this is arm, this is stage, this is base. And here is the light intensity control. Alright, we didn't know we had that. And here it is the substage condenser. From the name it is looking like substage, it is the part of stage. But always know that it is below the specimen and above the light, right? This is the mechanical stage, like you can rotate it or change. Like, you don't have to move your slide, but you can change the stage position, you know, move it or shake it or whatever you do with it. And at this side you can say, guys, I know I'm randomly reading it, I should be reading it, you know, one by one, but whatever. This area, this is coarse focus adjustment. Like, we know the coarse focus adjusts this stage. Like, when you move it, you can adjust it from here. Like, okay, we don't move it by hand, we move it from here. While this fine one, you know, change it or focus your eye lenses. Some microscopes have it here, right? Alright, here it is, interpupillary. Okay, so this is the eyepiece adjustment, the fine focus, while this one is the interpupillary. You know, some people's eyesight is different, right? So you can change it from here to make it okay with every eyesight. And it is not just about the eyesight. Usually, yeah, it's about eyesight. Like, if you are perfect, you have to make it to point to where you can see it perfectly fine. And stage adjustment knobs, aperture, diaphragm control. I don't know what the diaphragm control is. Maybe we can read it in the future. Base with a light micro source, objective lenses 4. It can be only one. Like, in our, you know, lower classes, we only usually have one magnification, and it can be more than 4, 5. It can be 5 or it can be 6. And yeah, that's all. What is written? It is a bright field, means the background is white. The part of a modern bright field microscope. The microscope picture is somewhat more sophisticated than those found in many student laboratories. Means they are saying that this one is advanced. For example, it is binocular, has 2 pieces, and has a mechanical stage, an adjustable substage condenser, and built-in illuminator. Yeah, they are saying that we are showing you an advanced image. So, oh, here look at the inner diagram, how the lighting works. Here the light is out. This is the reflection by mirror, I think, or maybe by lens. and it goes into a condenser. Look how the light is condensed into one point, and it goes to the specimen. Here is our objective lens, and the light goes into that, and there are the mirrors to change the trajectory of that if you want to move the eyepieces so nothing differs. And here if you look at this lens again, it changes the focal length of the light that came from the below objective lens and make it even more magnifiable to our eyes. Right. The light path in an advanced bright field microscope. We know how it works in the inside. The microscopic resolution, we are going to leave it for the next live. This introduction is enough. I think tomorrow is going to be difficult, while the dark field, the phase contrast. So tomorrow we are going to study, or tomorrow or another day, we are going to study the types of the light microscope. Right, finish it. Alright, we are ending.